Welcome back everyone to Research Works podcast here at the pre-conference workshop in Melbourne, EACD and IAACD 2025. And our guest today is someone I've been wanting to speak to for quite some time. It's quite a privilege, Professor Darcy Fehlings. Thank you so much for being here.
Oh, I'm delighted to be here. Thank you.
I was so delighted when you said yes to be on this podcast. It's always, one of those things that I think is strange. I suppose the person who publishes so much and has done so much work in the area. You have people like me that have read your work on the other side and have benefited from a lot of the work that you have published. Out of all the things you have done, as a bit of a starting point, what is the one thing that you feel like you're most proud of? It's a tough one. It's like choosing your favorite child in a way, isn't it? Sorry.
Well, thank you. As a clinician researcher, I try to chase the things in research that I think are important clinically. And so I'm going to answer in two parts.
Beautiful.
So
I love that.
One of them is just really identifying that pain is an important part of the story for children with cerebral palsy, bringing awareness to pain and the need to manage it. But linked to that, in my practice, I see a lot of children with dystonia, with cerebral palsy and pain. And so working to bring forward a clinical practice guideline to help clinicians around managing dystonia and cerebral palsy, I felt was important work.
Yeah. Oh, no, I think it's absolutely important. I remember reading that work as well because it is a group of children, while small, is significant and the impact is across the entire family. And pain is just so underreported to the point where it's almost so normalized. I remember after reading some of your work, there was a child that I've been working with for a long time and we just asked the question, do you have any pain? And he, as an 8-year-old, said, yes, my foot's always in pain.
I know.
Yeah.
One of our earlier studies on pain, we consecutively asked over 400 children.
Wow.
And we had 25% who said, yes, they were in pain to the point that it was restricting their activities. That was quite disheartening for me because these were children in our practice receiving what I felt was excellent care. I thought, there's a little bit of work to do.
Yeah, it's sobering, isn't it? When you come at it from a pure place of curiosity and you ask the question, these are the things that you do find, and they do change your practice and your perspective. It's quite incredible. So your work you're a clinical researcher, which more I realize is quite a rare commodity, to have people who have the capacity for both clinician work and research. It's hard. What's your current role at the moment?
It is as a clinician scientist. I feel very privileged and very happy to be in that role because I love being a clinician, but I also love doing clinical research. And working in Canada, I've had the opportunity to really try to do that clinical research and lead some translational neuroscience networks where we're really trying to foster research across a spectrum, from the basic translational all the way to implementation. Let's get these early interventions into practice.
Yeah, that's right. That's been really exciting seeing that whole global movement, really, isn't it? And everyone's producing a lot of content to be able to push the field forward, which is such a privilege to be able to watch and see. So with your work that you're presenting here at the conference, you're part of the early intervention day in the workshop. Your presentation really does intrigue me, and it's all about genetics and biomarkers and words that we're hearing a lot at the moment. Very buzzwords, but a very exciting area. Do you mind talking us through a little bit about what your presentation is about?
Absolutely. I'm really happy to talk about genetics because it's an actively evolving story in cerebral palsy. We're relatively late in the game, but recently identifying that there is a genetic contribution to etiologic risk of developing cerebral palsy. And why I think it's important is that we really need new interventions for individuals with cerebral palsy. We need to evolve the field forward. And understanding some of the basic mechanisms to what leads to cerebral palsy, in this case genetics, I think will help to drive some of that innovation in intervention eventually.
Interesting.
Yeah.
Do you have any examples of some of the findings so far that have really started to influence practice when it comes to genetic testing? And
we're a little far away from impacting clinical practice. However, I would say that I'm starting to see a shift in physicians who are part of the diagnostic process of, assessing, do I feel that a CP description or a CP diagnosis would fit this child? And I'm starting to see those clinicians say, in addition to doing a good clinical history, a good clinical physical examination, and an MRI or some brain imaging, should I do genetic testing? And we're starting to understand what are these phenotypic features that might point towards a genetic etiologic risk for cerebral palsy. So for example, if we do an MRI and the MRI is normal, that's a marker. Okay. Clinician, you should consider a genetic workup.
How difficult is it, or how easy is it to do a full genetic testing profile?
It is challenging, and it's a shifting landscape. So I'll give you an example of why I say it's challenging. In my clinical practice, I can order CP gene panels. In my research, I can do whole genome sequencing, which is going to be the gold standard. And so there's a big discrepancy between what's available clinically and what's available within the research paradigm, but it's shifting. And the cost of doing whole genome sequencing is really coming down. So at some point it will be the most cost-effective genetic test. And why that is important for cerebral palsy is that although we know some of the genes that contribute to CP that are on the gene panel, we don't know them all. We're adding genes all the time. And whole genome sequencing allows us to look across all of the architectural contributions of genomics to CP. It allows us to detect single nucleotide variants, copy number variants. And in our study that we recently published in Nature Genetics, we screened everybody for mitochondrial variants and found mitochondrial variants as well, which are not typically associated with the CP phenotype. So that's
what you mean by evolving.
Yes.
Right. I think in that paper, there was an 11.3, there were pathogenic variants in 11.3 of cases.
That's right. So in this particular study, which was a representative sample, so we took all children with cerebral palsy that we were able to enroll in the study. Many of the publications in the CP genetics world right now are more biased samples. They are looking at children who are labeled cryptogenic or idiopathic, where they don't have etiologic risk. We know that those, we're going to get higher diagnostic yield. So this is a population-based sample, which is important, that we found 11%. So if you think about it, 1 in 10 children are going to have some genetic contribution to their etiologic risk for cerebral palsy within all children with cerebral palsy. So that is fairly high. But here's also the evolving component. So in addition to that 11%, we had 18% that had variants of uncertain significance. That doesn't mean that they're not likely to contribute. It means they're likely to contribute. We just don't have all the details yet. And so if we add that component in, we're up to 35, 36%. So it's a big number.
It is. Oh, thank you for breaking that one down, because that is a very interesting number. in the rare disease space, I know that they talk a whole lot about the diagnostic odyssey and looking for causes and looking for a diagnosis. Do you think this work that you're looking at doing in the area of cerebral palsy will help with that? Because I suppose people often do look at scans, nothing showing up on the MRI. Would this help in that process in terms of coming to a diagnosis?
Definitely for that family that has gone along a path and there aren't identified etiologic risks within what I'm calling environmental risks. They're not born preterm. They haven't had neonatal encephalopathy.
Sure.
It will help there because the likelihood is that you're going to find something, particularly if you do whole genome sequencing. However, I just want to caution that we have to think about things in a complex way as it relates to genetic contribution, because in our data, we found that the majority of children who had a genetic pathogenic or likely pathogenic variant also had numerous environmental risks.
Oh, right.
So what we're seeing is this an interaction between gene and environment. And we need to actually study that more so that we can understand, what are the different genetic genes that might lead to a baby being more at risk for neonatal encephalopathy, and that's the interaction. What are the different genes that lead to a risk for hemorrhaging, for example?
Right. So I can see that this is very important. Would you say this is still early work and this is...
Yes, I do think we're early on in understanding the story. And so we have to be careful about research methodology, just awareness of the type of sample of children we're getting or individuals with cerebral palsy that we're getting information from. Making sure that we're working with good genetic colleagues who really understand, gene burden analysis and aspects like that. So it's a real team effort.
Is it from a training perspective in terms of who's part of the team and what forms the team? Do you see that changing a little bit in terms of who the multidisciplinary teams and around the child? Or a lot of these skill sets be within what we already have and we're just adding more to that?
I think within the diagnostic workup, there will be more need for genetic consultation. That will be more part of the clinical picture. And then within the clinical research component, we definitely need to add the genetic basic scientists who really have an in-depth knowledge of how to look at genes and understand different research methodologies. So for example, there is an AI tool now called AlphaFold that I personally hadn't heard about. And we can use that AI app tool to predict the pattern of the gene impacting how the protein is going to form and how predict whether it will be deleterious or not. So there's different knowledge base and tool sets that we need to have access to.
So what do you think, how far away do you think all of this is from a where do you see this going, I suppose?
We're really, it's actively exploding right now. So we're on the rapid upturn. What's harder for me to predict is how long it will take to get to that goal of influencing and developing new interventions based on the genetic treatment.
It's always good to know about what's evolving, what is around, and I'm just so grateful you've been able to clarify the current status and where we're headed. And I'll definitely be keeping an eye out on where this is all going, Darcy.
Thank you.
What's next for you in terms of your research and your research team? Are you mainly focused in this area, or you still have a lot of different areas that you're involved with?
We definitely have a strong focus on genetic contribution within our study called CPNet. we have a strong genomics platform. So we'll continue to move that forward.
Wonderful.
And then from overall research projects, I'm shifting a little bit to implementation at this point.
I love that.
Trying to drive actual change of practice and things that I think are important. So I'll definitely continue to do that as well.
Brilliant. And then it's wonderful hearing that. We spoke with Alicia Spittle a few months ago, and I said the same thing to her. I said, where to? She's like, I think right now we just need to implement. We have so much knowledge. We've gained so much in the last decade or even 5 years. And we're at that place now where we just need to implement.
I'm really feeling the call of implementation as well.
That's fantastic, fantastic. Yes. Well, thank you so much for joining us today. I really appreciate your time, Darcy, and for sharing your knowledge here at the pre-conference workshop.
I really enjoyed talking with you as well.
Wonderful. Thank you so much. Thank you for being part of that as well. I hope you got as much out of that as I did, and to hear the current status of what we're looking at right now in cerebral palsy. So stay tuned for so much more. We'll talk to you all again really soon. Bye.